Hello everyone, so we are talking about purine nucleotide synthesis, and this is a very important topic. So how do we eventually get a purine nucleotide via biosynthesis? There are two ways of going about this. First of all is de novo synthesis. In de novo synthesis, the purine nucleotides are produced in the body from scratch. There is also another pathway known as the salvage pathway. In the salvage pathway, we can say that the adenine and the guanine are salvaged. Now, this might be a bit of a hard thing for you to understand because of some basic things which need to be understood first. So, I'll begin with that. When we talk about the word purine, let's start with the term purine bases. The nitrogenous bases found in the human body can be divided into the purines and the pyrimidines. What do you understand about purine and what do you understand about pyrimidines? So purines are the bases that have two rings, whereas the pyrimidines are the ones that have one ring. In the human body, in case of the ribonucleotides, we have, and even in the case of the deoxyribonucleotides, we have two purine bases, adenine and guanine, right? And also we have, that's basically for the bases. Now the purine bases, whenever they combine with a sugar, a pentose sugar, they can form either ribonucleotides and they can form deoxyribonucleotides. They can form ribonucleosides and they can form deoxyribonucleosides, right? So the difference between a base and a nucleoside is simply the addition of a pentose sugar, and we have two types or two varieties or two options, and that you will understand the very fine difference between the formation of the ribonucleosides and the deoxyribonucleosides when we go over this chapter as a whole, which I'm hoping I'll be able to complete in this one video. So the nucleosides basically include adenosine and guanosine. Then we talk about the nucleotides, which include phosphate, and we can have 1-phosphate, 2-phosphate or 3-phosphate. All three are examples of nucleotides. So we write adenosine monophosphate, guanosine monophosphate. We might also write adenosine diphosphate and adenosine triphosphate. So I'm hoping you've got the basics down. So when we talk about the synthesis of the purines, what are the basic raw material that we absolutely need? Going back to what we were saying about purine nucleotide synthesis, and we know we absolutely need the nucleotides for DNA and RNA formation. So I said that there are two ways of going about this. Either there is de novo synthesis or there is salvage pathway. In de novo synthesis, the adenosine monophosphate and the guanosine monophosphate, which are to be integrated in DNA or RNA, they are basically produced from scratch. Produced from the beginning in the absence of any set precursor. And that is one way of doing it. And that is the major pathway. And there's also the salvage pathway. In the salvage pathway, the bases, for example, adenine or guanine, which enter the body simply because they were consumed. For example, a person ate meat or person ate some plant, and obviously the DNA material is there as well, and that will similarly be consumed and partially digested. Or sometimes simply the recycling of DNA and RNA leaves behind some bases which can be shuttled into the salvage pathway, and the nucleotides can be produced again to be integrated into DNA. So this is a major difference. In de novo synthesis, we are producing the purine nucleotides from scratch. And what are the things that we need for that? We need the integration of the amino acids, 3 amino acids. We need aspartate, we need glycine, and we need glutamine. So first of all, you need to remember that for this de novo synthesis, you need 3 amino acids, aspartate, glycine, and glutamine. Aspartate, glycine, and glutamine. You will also need CO2 because there will be an important carboxylation in one particular step, and you will also need N10-formyl THF for in 2 steps, I believe. THF is basically the active form of a vitamin B9, which has the most important function of being a 2-carbon carrier, and in this case the 2-carbon group that is carried is the formyl group. So these amino acids, aspartate, glycine, glutamine, will be contributing mostly nitrogen. The CO2 and the formyl group will be contributing the carbon. Now all of these carbons and the nitrogens derived from aspartate, glycine, glutamine, CO2 and the N10-formyl THF molecules will be added to a pre-existing molecule of ribose 5-phosphate. So you must never forget this, and all of this will be happening where? In the liver, interestingly. That is something you must not forget. Now we will talk about the actual steps. So in the first step we have we'll be producing PRPP. Now PRPP basically stands for phosphoribosylpropyl. Actually, more accurately, it stands for 5, because that is the positioning. 5-phosphoribosyl 1-pyrophosphate, right? This is a very interesting molecule, and in the first step we will have the formation of this. This will be involved not only in de novo synthesis but also in the salvage pathway. So I'm hoping this is a bit of a clarification of its importance. So the question can be, what exactly is 5-phosphoribosyl pyrophosphate? We can say that it is an activated pentose. Why? Because of the addition of this portion over here. So this is an activated pentose. And how do we form this? We start with, as I've previously mentioned, a molecule of ribose 5-phosphate, which is basically a ribose pentose sugar with a phosphate at the 5th position, and you activate it basically by a reaction that goes something like this, and you end up with the formation of PRPP. The name of the enzyme is relatively easy. It is PRPP synthetase, right? You start with ribose 5-phosphate and you end up with 5-phosphoribosyl pyrophosphate. You can already conclude that in this particular step, the only thing that has been added is 1 pyrophosphate molecule to this particular molecule over here. This and this is the same, so we've simply had the addition of a pyrophosphate. The name of the enzyme is easy enough to remember. It is simply PRPP synthetase, and whenever we use the synthetase enzymes, ADP is always needed. That is not a new concept, so we have to show that ADP is consumed in the very first step. So that is how you go about showing this. So this is the first and very important reaction. In this reaction, to reiterate, simply we've started with the ribose 5-phosphate. We have converted it into an active phosphate, into an active pentose, which is 5-phosphoribosyl 1-pyrophosphate. So we've had the addition of a pyrophosphate moiety to ribose 5-phosphate in order to produce PRPP. And the name of the enzyme is simply PRPP synthetase because we have synthesized it. And whenever synthetase enzymes are used, ADP is consumed because it is an energy dependent reaction. And because we've had the addition of a pyrophosphate, this means that the ADP molecule in question has been degraded to AMP and not to ATP, as is the usual case. This is a very important reaction. The thing that must not be forgotten is that in addition to the ADP that is consumed and the AMP that is produced from its consumption, we also require a cofactor, which must not be forgotten is magnesium, right? So that is a very important cofactor for the first step. Now let's talk about the allosteric regulation of this step, which is not as important as for the next one, but regardless very important. Inorganic pyrophosphate is going to act as the allosteric activator, whereas the AMP and the GMP, which are going to be the end products of this reaction, will act as the allosteric inhibitor. So this, for example, this over here is an example of end product inhibition. Inorganic phosphate is the allosteric activator, and the AMP and the GMP are the allosteric inhibitor. So this is the first reaction, very important. The next step is similarly of extreme importance, and in this one we simply have the reaction between 5-phosphoribosyl 1-pyrophosphate and we have this really interesting reaction to form 5-phosphoribosyl. This part is the same. This reaction is not any 5-phosphoribosylamine. So we've had what we would simply consider a substitution, a mere substitution, right? So take a look at this. We've simply had this substitution. Actually, when we talked about all of this, I did not specify the amounts that were needed. In this reaction, we will need in total 2 molecules of glutamine. This is the number, and they will be required in step number 2 and in step number 5. And right now we are doing step number 2. So as usual, in my blue color, I'll be showing you the incoming glutamine like this. Glutamine is incoming, and then we'll have the formation of glutamate. And the difference between glutamine and glutamate is that this will have 2 NH2s and this one will have just a single because of the donation. So the interesting thing is the name, and the name of this enzyme is not going to be very hard. It is going to simply be glutamine, because that is one of the reactants. Glutamine phosphoribosyl phosphate, which is the name of the reactant. Phosphoribosyl pyrophosphate, amidotransferase, because in between these 2 reactants there will be the transfer of an amido group from glutamine to phosphoribosyl pyrophosphate, and because of this transfer we will have the formation of an amine group in the over here. So this is basically this is basically the second reactant. This one is the first, which we've talked about. This is the reactant 5-phosphoribosyl 1-pyrophosphate. This is the second reactant, glutamine 5-phosphoribosyl pyrophosphate, and the reaction is an amido transfer. An amido transfer from where exactly? From the glutamine to the PRPP, and we've had the formation of this molecule over here. So this is the rate limiting step. This step is very important. The second step is very important because it is a rate limiting step, the rate limiting step. And you must be very careful in remembering that the allosteric activator in this case is going to be the substrate PRPP, and this will have its importance later on, PRPP. And the inhibition will once more be end product inhibition. AMP and GMP will be allosteric inhibitors. So we have substrate activation, and end product inhibition. It's not that hard to remember if you try to keep it in mind. Substrate activation and end product inhibition.